Hello, welcome to the JASN podcast. I am Katalin Susztak. I'm one of the associate editors of JASN. Each month in the JASN podcast, the editors talk about one or more papers in the current issue, often with one of the authors. Our hope is that this discussion will prompt you to go to our website or turn to the latest issue that is now in your mailbox. It's a great honor to introduce you to today's paper. Joining me today is Susan Quaggin. Dr. Quaggin is the Chief of Nephrology and Professor of Medicine at Northwestern University. Susan also recently completed a term as a president of the American Society of Nephrology. Susan, thanks for joining me for this conversation. I suspect you are glad to have a bit more time to devote to your science. JASN is glad to be publishing this interesting study.
Well, thank you, Katalin. It's a real honor for me to be here with you, and I'm just delighted.
So the paper we're going to talk about today is entitled Activation of Angiopoietin-Tie 2 Signaling Protects the Kidney from Ischemic Injury by Modulating of Endothelial-Specific Pathway. The first author of the study is Yi-Yang Li, and you, Susan, are the senior and corresponding author. So first, let's start with ischemic injury. Why is this an important topic?
Well, terrific question, Katalin, and I think both of us are nephrologists and we see patients in the hospital, and it's absolutely clear that ischemic acute kidney injury is still incredibly common, and despite it being one of the commonest reasons why we're consulted as nephrologists in the hospital, we really have limited treatments for this serious condition. So this is why we were very interested in trying to identify both new molecular pathways, but also ultimately to identify some new candidate therapeutic targets so that we might intervene and help these patients in the hospital.
Well, I can affirm that. I was just coming off service this morning, and then we had a case of APN that we had to do dialysis this weekend. So due to, vascular changes, indeed, it remains a very important issue. So how about the blood vessels? What do we know about angiopoietin and Tie 2? I know you have been working on this pathway for the last decade. So can you tell us quickly about, what should we know about angiopoietins, ties, and the vessels?
Sure. Thanks for the question. And yes, we've been working on it, I think, maybe 15 years or even longer. And the reason we got interested in this particular vascular growth factor signaling pathway is that there were a lot of papers published showing associations of dysregulation of this pathway in a number of serious clinical vascular diseases, including acute kidney injury due to ischemia. So just sort of an overall view of this pathway, I like to say that activation of angiopoietin-Tie signaling promotes happy and healthier blood vessels. And it does this by actively promoting quiescence of the vessels, promoting survival of blood endothelial cells, reducing their responsiveness to inflammation, and promoting endothelial junction stability, so reducing capillary leak. And if you think about all of those things, we know from a lot of other wonderful work in acute kidney injury due to ischemia that all of these functions of endothelial cells are disrupted. So that's why we were interested in the Ang-Tie pathway and the idea really to boost this signaling pathway to protect blood vessels and in the injured kidneys.
Sounds very important. Is there a difference between Tie 1 and Tie 2? It seems you have focused on Tie 2 and a signaling pathway called VEPTP, vascular endothelial protein tyrosine phosphatase.
Yes, so great question. And I will say to this day, we are not entirely sure about all of the roles of Tie 1 versus Tie 2, but Tie 1 is related to the Tie 2 receptor. They're both expressed as membrane tyrosine kinases by endothelial cells. But unlike Tie 2, Tie 1 is an orphan receptor, so it can't bind the angiopoietin ligands. The angiopoietins are sort of like the key that goes into the lock, if you think about Tie 2. And when they bind to Tie 2, they result in changes, including phosphorylation, which leads to signals within the cell, and a whole number of these pathways are activated that protect the blood vessels. So we focused on Tie 2 because you really need to boost its activity in order to change a cell's behavior and to result in this protection of the blood vessels. You also mentioned another molecule. So this pathway becomes a little bit complicated because there's multiple players, but there's a really interesting endothelial specific phosphatase, which, as you mentioned, is called VEPTP. And what it does, it actually can physically interact with the Tie 2 receptor, and it deactivates it almost like a light switch, turning it off. It rips off all the phosphates so that the signaling is dampened. And the reason that we were interested in looking at that, we knew that that phosphatase is very regulatable. So its expression goes way up in hypoxia. And if we think about ischemia reperfusion injury in the kidney, we know it's a hypoxic injury to the kidney. And sure enough, when we looked for this phosphatase, levels of it go way up in the blood vessels in the injured kidney. So this seemed like a really good foothold to start to understand this pathway and start to try to modulate it to protect the kidney.
Oh, that sounds very exciting. So is that what prompted you to study this specific pathway or perform these experiments?
Yes, I think this, and also the fact that there was a lot of data out there that suggested that elevated levels of what used to be thought of as an inhibitory angiopoietin, angiopoietin-2, is linked to bad outcomes or worsened kidney injury following ischemia reperfusion. So that came from patient studies, came from animal model studies, but there was a problem with it in that angiopoietin-2 sometimes is an antagonist, sometimes an agonist, so it's complicated. And so we were really interested in trying to dive down and understand all of the players in the pathway and figure out where we could actually intervene. At what point in this pathway could we intervene to protect the kidneys? And that's what we did throughout the paper, and really kind of were able to figure out, following ischemia reperfusion, why Tie 2 is switched off, almost like a light switch. And we feel it's really due to this increased expression of the phosphatase, which in turn downstream leads to increased levels of angiopoietin-2 in the bloodstream, which is still a very good biomarker, but might just be a surrogate of what's really going on and what's causal here. So complicated pathway, but I think we have a much better understanding now, and it's exciting that we might really be able to leverage these findings to develop some new candidate treatments.
Oh, I see. So let me try to sum up. So angiopoietin-2 is a bad guy. Increased level of angiopoietin-2 has been associated with bad outcome. And the VEPTP is a phosphatase inside the endothelial cell. Its level goes up in ischemia, and Tie 2 has maybe a kind of complex regulation because it seems activated by Ang2, but the VEPTP is turning its level off. Is that how we could sum up?
Almost pretty close. It seems that angiopoietin-2 is a sometimes can activate and sometimes can inhibit Tie2, and it depends on whether there's inflammation around. So it is complex, but what we show in the paper really is that following an ischemic injury to the kidney, this VE-PTP that's in blood vessels goes way up. That turns Tie2 off, and that actually also results in a transcription factor staying within the nucleus and increasing transcription of angiopoietin-2. So it's more of a complicated pathway, but it really seems that VPTP may be the reason why we're seeing these elevated levels of angiopoietin-2 that are being elevated transcriptionally following turning off Tie2. So it's like a feedback loop that's happening within the blood vessels. The good news is that I think once we get to developing a treatment, and in the paper we use two ways to turn that Tie2 receptor back on, almost like turning the light switch back on in the blood vessels. We do it by either inhibiting the phosphatase or giving back the ligand angiopoietin-1, which is a really potent activator, and we've developed a new biologic. So I think the good news is, we can think about treatment without actually having to worry so much about all of the complexities of the signaling pathway when it ultimately makes it through to later preclinical studies and then ultimately, as we hope, into the clinic.
That is absolutely fantastic. So can you guide us through just a couple of sentences? What are the tools that you used in this study to understand the complexity of this pathway, and how did you come up with this novel therapeutics?
Yeah, so as you might see, if any of the listeners go and look at the paper, you'll see a large group of names of co-authors. So really a phenomenal team of individuals, trainees, clinical fellows, scientists who I have the privilege of collaborating with. So a large group who, together we developed new genetic mouse models. We utilize some, types of experiments that you have really pioneered, so single cell and RNA-seq. Also, this new biologic that was really developed by two brilliant scientists here at Northwestern University, Pan Liu and Jing Jin, who designed this new angiopoietin-1 mimetic that is long-lasting after it's injected and is super potent. And in order to do that, they had to do some really smart design of the molecule, and we've used it in a number of different vascular diseases, as have they, and it's now actually in development for clinical use by a biotech company in Canada. So clearly a team effort and lots of different methodologies used in the paper to explore and try to understand, how we can leverage this pathway to help protect kidneys.
Wow, that is very, very impressive. So this is called the hepta-ANG1 that now we could give to mice in this paper, which seem to improve AKI or ischemia in mouse models. And what you're saying that this particular biological, I believe, right, is also in clinical or potentially to be ready for clinical trials for patients.
Yes. So I think absolutely the hepta-ANG1 works great in mice. It's actually a human hepta-ANG1 and ultimately could work in other sorts of animal models as we think about development of something for the clinic. Clearly, there's a lot more work that needs to be done, but it is in development by a biotech company, and it's exciting to see, and we'll just have to wait and see. I will say there's other groups as well around the globe who are doing wonderful work on vascular signaling pathways in Angiopoietin-Tie, and there are other potential therapeutics in this space that also might make it into the clinic. So I think it's exciting to see where this might lead.
So it seems that there are multiple pathways that we could potentially intervene in endothelial cells to improve their function and make them less leaky in the context of ischemia. And what you're saying is that will protect from ischemic reperfusion injury hopefully in patients as well.
Yep, that would be the dream, I think. And clearly lots to do, but I think it's really exciting times. And I think, certainly from your work as well, I think we're learning so much also from some of these models where we can now apply, newer ways to drill down with single cell and other ways. So, so we're hopeful, and certainly this pathway, I think, will play an important role in therapeutics in a variety of different vascular diseases, including ischemia reperfusion injury.
This is super exciting, and then we can't wait to see this new therapies which we really need for ischemia translated to patients. Is there anything else you would like to add to the discussion?
No, just to reiterate what a pleasure it is to be here and get an opportunity to speak with you as well. And again, just a huge debt of gratitude to all of the team members who made that paper possible, and a huge thank you to JASN for publishing it.
It is a fabulous paper. I really would like to make sure that the audience will have the opportunity to read them online or maybe in print. It seems that this could lead to novel therapies that we could hopefully translate to patients, which we badly need for ischemia reperfusion, a condition that we see in the hospital. So I would like to thank you for spending time with us and publishing with JASN, and we can't wait to see the extension of these studies, and hopefully one of them is going to be translating these findings from mice to patients. And thank you so much again.
Great. Thank you, Katalin.
This podcast is copyrighted by the American Society of Nephrology. All rights reserved. All content in this podcast is for informational purposes only and is not intended to be medical advice. This podcast should not be used in a medical emergency or for the diagnosis or treatment of any medical condition. Please consult your doctor or other qualified healthcare professional if you have any questions about any medical condition or before taking any drug, changing your diet, or commencing or discontinuing any course of treatment. Thank you for listening to this podcast from the American Society of Nephrology.